B8. Chloroplasts. Chloroplasts, the organelles responsible for phagocytosis, are in many respects similar to mitochondria. Both chloroplasts and mitochondria function to generate metabolic energy, evolve by endosymbiosis, contain their own genetic systems, and replicate by division. However, chloroplasts are larger and are more complex than mitochondria, and they perform several critical tasks in addition to the generation of ATP. Most importantly, chloroplasts are responsible for the photosynthetic conversion of CO2 to carbohydrates. In addition, chloroplasts synthesize amino acids, fatty acids, and the lipid components of their own membranes. The reduction of nitrate NO2 to ammonia NH3, an essential step in the incorporation of nitrogen into organic compounds, also occurs in chloroplasts. Moreover, chloroplasts are only one of several types of related organelles, plastids, that play a variety of roles in plant cells. The structure and function of chloroplasts. Plant chloroplasts are large organelles, 5 to 10 millimeter long, that, like mitochondria, are bounded by a double membrane called the chloroplast envelope. In addition to the inner and outer membranes of the envelope, chloroplasts have a third internal membrane system called the thylakoid membrane. The thylakoid membrane forms a network of flattened discs called the thylakoids, which are generally arranged in stalks called grana. Because of these three membrane structure, the internal organization of chloroplasts is more complex than that of mitochondria. In particular, there three membranes divide chloroplasts into three distinct internal compartments. 1. The internal membrane. The intermembrane spaces between the two membranes of the chloroplast envelope. 2. The stroma, which lies inside the envelope. 2. But outside the thylakoid membrane. And 3. The thylakoid lumen. Despite these greater complexities, the membranes of chloroplasts have clear functional similarities with those of mitochondria, as expected, given the roles of both organelles in the chemiosmotic generation of ATP. The outer membrane of the chloroplast envelope, like that of mitochondria, contains porins and is therefore freely permeable to small molecules. In contrast, the inner membrane is impermeable to ions and metabolites, which are therefore able to enter chloroplast only via specific membrane transporters. These properties of the inner and outer membranes of the chloroplast properties of the inner and outer membranes of the chloroplast envelope are similar to the inner and outer membranes of mitochondria. In both cases, the inner membrane restricts the passage of molecules between the cytosol and the interior of the organelle. The chloroplast stroma is also equivalent in function to the mitochondrial matrix. It contains the chloroplast genetic system and a variety of metabolic enzymes, including those responsible for the critical conversion of CO2 to carbohydrates, photosynthesis. The major difference between chloroplast and mitochondria in terms of both structure and function is the thylakoid membrane. This membrane is of central importance in chloroplast, where it fills the role of the inner mitochondrial membrane. In electron transport and chemiosmotic generation of ATP, the inner membrane of the chloroplast membrane envelope, which is not folded into crystal, does not function in photosynthesis. Instead, the chloroplast electron transport system is located in the thylakoid membrane, and protons are pumped across the membrane from the stroma to the thylakoid lumen. The resulting electrochemical gradient then drives ATP synthesis as protons cross back into the stroma. In terms of its role in generation of metabolic energy, the thylakoid membrane of chloroplast is thus equivalent to the inner membrane of mitochondria. The chloroplast genome. Like mitochondria, chloroplasts contain their own genetic system, reflecting their evolutionary origins from photosynthetic bacteria. The genomes of chloroplasts are similar to those of mitochondria in that they consist of circular DNA molecules present in multiple copies per organelle. However, chloroplast genomes are larger and more complex than those of mitochondria, ranging from 120 to 160 kb and containing approximately 120 genes. Both the ribosomal and transfer RNAs used for translation of chloroplast mRNAs are encoded by the organelle genome. These include 4 RNAs, rRNAs 23S, 16S, 5S, and 4.5S, and 30 tRNA species. In contrast to the smaller number of tRNAs encoded by the mitochondrial genome, the chloroplast tRNAs are sufficient to translate all the mRNA codons according to the universal genetic code. In addition to these RNA components of the translation system, the chloroplast genome encodes about 20 ribosomal proteins, which represent approximately a third of the proteins of chloroplast ribosomes. Subunits of RNA polymerase are also encoded by chloroplast. All the additional RNA polymerase subunits and other factors needed for chloroplast gene expressions are encoded in the nucleus. The chloroplast genome also encodes approximately 30 proteins that are involved in photosynthesis, including components of photosystem 1 and 2 of the cytochrome BF complex and of ATP synthase. In addition, one of the subunits of ribulose biphosphate trans-carboxylase, Rubisco, is encoded by chloroplast DNA. Rubisco is the critical enzyme that catalyzes the addition of CO2 to ribulose 1,5-biphosphate during the carbon cycle. Not only is it the major protein component of the chloroplastoma, but it is also thought to be the single most abundant protein on Earth, which Rubisco. So it's noteworthy to note that one of its subunits is encoded by the chloroplast genome. And the membrane-enclosed organelles, as discussed under structure and function of cytoskeleton and its role in motility, discussed in the preceding section, constitute one level of organizational structure of eukaryotes. A further level of organization is provided by the cytoskeleton, which consists of a network of protein filaments extending throughout the cytoplasm of all eukaryotic cells. The cytoskeleton provides a structural framework for the cell, serving as a scaffold that determines cell shape and the general organization of the cytoplasm. In addition to playing the structural role, the cytoskeleton is responsible for cell movements. These include not only the movements of entire cells, but also the internal transfer of organelles and other structures, such as mitotic chromosomes, through the cytoplasm. Importantly, the cytoskeleton is much less rigid and permanent than its name implies. Rather, it is a dynamic structure that is continually reorganized as cells move and change shape, for example, during cell division. The cytoskeleton is composed of 3 principal types of protein filaments: actin filaments, intermediate filaments, and microtubules, which are held together and linked to subcellular organelles and the plasma membrane by a variety of accessory proteins. This chapter discusses the structure and organization of each of these 3 major components of the cytoskeleton, roles in cell motility, organelle transport, as well as their roles in cell motility, organelle transport, cell division, and other types of cell movements. Structure and organization of actin filaments. The major cytoskeletal protein of most cells is actin, which polymerizes to form actin filaments. Thin flexible fibers approximately 7 nanometers in diameter and up to several micrometers in length within the cell. Actin filaments, also called microfilaments, are organized into higher order structures forming bundles of 3-dimensional networks with the properties of semi-solid gels. The assembly and disassembly of actin filaments, their cross-linking into bundles and networks, and the association with other cell structures such as the plasma membrane are regulated by a variety of actin-binding proteins, which are critical components of the actin cytoskeleton. Actin filaments are particularly abundant beneath the plasma membrane, where they form a network that provides mechanical support, determines cell shape, and allows the movement of the cell surface, thereby enabling cells to migrate, engulf particles, and divide. Individual actin molecules are globular proteins of 375 amino acids, 43 kD. Each actin monomer, globular G actin, has tight binding sites that mediate head-to-tail interactions with 2 other actin monomers. So actin monomers polymerize to form filaments, filamentous F actin. Each monomer is rotated by 166 degrees in the filaments, which therefore have the appearance of a double-stranded helix Because of all the actin monomers oriented in the same direction, actin filaments have a distinct polarity, and their ends, called the plus and minus ends, are distinguishable from one another. It is noteworthy that several drugs useful in biology, cell biology, act by binding to actin and affecting its polymerization. For example, the cytochalations bind to the plus ends of actin filaments and block their elongation. This results in changes in cell shape as well as inhibition of some types of cell movements, for example, cell division following mitosis, indicating the actin polymerization is required for these processes. Another drug, phalloidin, binds tightly to actin filaments and prevents their dissociation into individual actin molecules. Phalloidin, labeled with the fluorescent dye, is frequently used to visualize actin filaments by fluorescence microscopy. Intermediate filaments. Intermediate filaments have a diameter of about 10 nanometers, which is intermediate between diameters of the two other principal elements of the cytoskeleton. Actin filaments about 7 nanometers and actin microtubules about 25 nanometers. In contrast to actin filaments and microtubules, the intermediate filaments are not directly involved in cell movements. Instead, they appear to play basically a structural role by providing mechanical strength to cells and tissues. Intermediate filament proteins. Whereas actin filaments and microtubules are polymers of single types of proteins, actin and tubulin respectively, intermediate filaments are composed of a variety of proteins that are expressed in different types of cells. More than 50 different intermediate filament proteins have been identified and classified into 6 groups based on similarities between the amino acid sequences. And types 1 and 2 consist of 2 groups of keratins, each consisting of about 15 different proteins, which are expressed in epithelial cells. Some type 1 and type 2 keratins, called hard keratins, are used for production of structures such as hair, nails, and horns. The type 3 intermediate filaments proteins include pimentin, which is found in a variety of different kinds of cells, including fibroblasts, smooth muscle cells, and white blood cells. And other types of protein, desmin, is specifically expressed in muscle cells where it connects with Z discs of individual contractile elements. Type 4 intermediate filament proteins include the 3 neurofilament NF proteins designated NF-L, NF-M, and NF-H for light, medium, and heavy, respectively. These proteins form the major intermediate filaments of many types of mature neurons. The type 5 intermediate filament proteins are the nuclear lamins, which are found in the most eukaryotic cells rather than being part of the cytoskeleton, and nuclear lamins are components of the nuclear envelope. They also differ from other intermediate filament proteins in that they assemble to form an orthogonal meshwork underlying the nuclear membrane. Despite considerable diversity in size and amino acid sequence, the various intermediate filament proteins share a common structure organization. Microtubules. Microtubules, the third principal component of the cytoskeleton, are rigid hollow rods, approximately 25 nanometers in diameter. Like actin filaments, microtubules are dynamic structures that undergo continual assembly and disassembly within the cell. They function both to determine cell shape and in a variety of cell movements, including some forms of cell locomotion and the intracellular transport of organelles and the separation of chromosomes during mitosis. Structure, assembly, and dynamic instability of microtubules. In contrast to intermediate filaments, which are composed of a variety of different fibrous proteins, microtubules are composed of a single type of globular protein called tubulin. Tubulin is a dimer consisting of 2 closely related 55 kD polypeptides, alpha tubulin and beta tubulin. Like actin, both alpha and beta tubulin are encoded by small families of related genes. In addition, a third type of tubulin, alpha tubulin, is specifically localized to the centrosome, where it plays a critical role in initiating microtubule assembly. Tubulin dimers polymerize to form microtubules, which generally consist of 13 linear protofilaments assembled around a hollow core. The protofilaments, which are composed of head-to-tail arrays of tubulin dimers, are arranged in parallel. Consequently, microtubules, like actin filaments, are polar structures with 2 distinct ends, a fast-growing plus end and a slow-growing minus end. This polarity is an important consideration in determining the direction of movement along microtubules, just as the polarity of actin filaments defines the direction of myosin movement. Tubulin dimers can depolymerize as well as polymerize. Tubular dimers can polymerize as well as polymerize, and microtubules can undergo rapid cycles of assembly and disassembly. Both alpha and beta tubulin bind GDP, which functions analogously to the ATP bound to actin to regulate polymerization. In particular, the GDP bound to B tubulin, though not that bound to alpha tubulin, A tubulin, is hydrolyzed to GDP during or shortly after polymerization. Whether a microtubule grows or shrinks is determined by the rate of tubulin addition relative to the rate of GDP hydrolysis. On to the next topic, C1. That's subsection C1, organization of genes. In molecular terms, a gene commonly is defined as the entire nucleic acid sequence that is necessary for the synthesis of a functional polypeptide. According to this definition, a gene includes more than the nucleotides encoding the amino acid sequence of a protein. Referred to as the coding region. A gene also includes all the DNA sequences required for synthesis of a particular RNA transcript. This diagram you see here shows that this is a typical eukaryotic gene. A gene structure for prokaryotic genes usually contain uninterrupted coding sequences, whereas coding sequences of eukaryotic genes are commonly interrupted by non-coding sequences. Thus, a gene is the entire DNA sequence required for synthesis of a functional protein or RNA molecule. In addition to the coding regions exons, a gene includes transcription control regions and sometimes introns, although the majority of genes encode proteins. Some encode tRNAs, rRNAs, and other types of RNA. Most bacterial genes have no introns, whereas most genes in the multicellular organisms do. The introns in human genes encoding average-sized proteins are often much longer than the exons. Many bacterial proteins with related functions are encoded by contiguous genes regulated by a single transcriptional control region. This type of gene cluster, called an operon, is transcribed into a single polycistronic mRNA, which is translated to yield several different proteins. Most eukaryotic transcription units are transcribed into monocistronic mRNAs, each of which is translated into a single protein. The primary transcript produced from a simple eukaryotic transcription unit is processed into a single type of mRNA. The primary transcript produced from a complex eukaryotic transcription unit can be processed into two or more different mRNAs depending on the choice of splice sites and or polyadenylation site. In the case of many complex units, one mRNA is produced in one cell type while an alternate mRNA is produced in a different cell type. Organization of genes and non-coding DNA. The genomes of prokaryotic and lower eukaryotes contain few non-functional sequences, whereas vertebrate genomes contain many sequences that do not code for RNAs or have any structural or regulatory function. Only about 5% of the genome DNA in a human encodes proteins or functional RNAs. The genomes of prokaryotes and lower eukaryotes contain few non-functional sequences, whereas vertebrate genomes contain many sequences that do not code for RNAs or have any structural or regulatory function. Only about 5% of the genomic DNA in human encodes proteins or non-functional RNAs. The lack of a consistent relationship between the amount of DNA, the haploid chromosomes of an animal or plant, and its phylogenetic complexity is called the C-value paradox. The human genome. This term is used to describe the different types of sequences that together make up the DNA in a human cell. The DNA of the human genome is about 3 billion base pairs pp long and is estimated to contain 30,000 genes. The DNA is arranged as a set of 23 chromosomes, each of which is a single double-stranded DNA molecule, 55 to 250 million pp long. The genes are gen-related sequences account for about 25% of the DNA. The remainder is called extrogenic DNA and not known function. Genes and gene-related sequences. 20 to 30%. The genes, the coding information in a gene is present as a series of segments of DNA sequence called exons, separated from each other by intervening non-coding sequences called introns. Genes vary greatly in size and also with respect to the number and sizes of the introns. Some genes, such as the histone H4 gene, are just a few hundred base pairs long. Others, such as the factor VIII gene, are several hundred kilo base pairs kbp in length and contain mainly large introns, such that the actual coding sequence accounts for just a few percent of the total gene sequence. Additional sequences are present which are associated with genes. Leader and trailer sequences occur at the 5 and 3' ends of the gene, which are transcribed but not translated. Promoter sequences occur upstream of the point where transcription begins in the regular synthesis of mRNA from the gene. The promoter may extend up to 1 kbp upstream, but other regulatory sequences that influence transcription may occur at sites much farther away. Gene family and some gene families. Some genes exist as a number of copies with identical or related sequences that can be grouped into families. Gene families may be organized in a number of ways. All of the genes in the family occur at the same chromosomal locus. An example of this is the growth hormone gene family, whose 5 members are clustered on chromosome 17. The genes belonging to the family may occur at different loci. For example, the 5 members of the aldolase gene family are on different chromosomes. The genes of a family may exist as a series of clusters on different chromosomes. An example is the home box genes, which occurs as 4 clusters on separate chromosomes, each containing about 10 individual genes. In some multigene families, all the genes are identical and may encode a protein required in large amounts by the cell, such as histones. In other families, the genes are not identical but show some sequence divergence. In some cases, the divergence is so great that the genes encode proteins that are related to but have distinct properties. An example of this is the alpha and beta globin gene families, whose members are expressed at different sites of embryonic development. Pseudogenes. These are diverged members of gene families that have acquired one or more inactivating mutations so that they are no longer able to function and do not produce biologically active protein. A pseudogene is simply a mutated version of a parent gene. Often the mutations present are nonsense mutations, which generate stop codons and result in premature termination of translation. Related to pseudogenes are processed pseudogenes, which are DNA copies of mRNA. Processed pseudogenes lack introns, and because they do not have a promoter, they are not transcribed and do not result in protein production. It is not clear how processed pseudogenes arose in the human genome, but they may have been derived by reverse transcription of mRNA into double-stranded DNA, which was then inserted into the genome. Another group of inactive genes are gene fragments which lack the 5 or 3' region of the parent gene. These are thought to have arisen by a deletion event or by a recombination that split the parent gene. Extragenic DNA, 70 to 80%. This part of the human genome is composed of sequences that exist in condition to the genes and gene-related sequence described above. In the figure shown, extragenic DNA is composed of sequences that are not part of a gene, exons and introns, not associated with a gene, leader and trailer sequences, promoters and distant regulatory elements, and not a pseudogene or a gene fragment. Extragenic DNA is composed of sequences that are not part of a gene, exons and introns, not associated with the gene, leader and trailer sequences, promoters and distant regulatory elements, and not a pseudogene or a gene fragment. Although extragenic sequences account for most of the DNA in the human genome, 70 to 80%, they have no known function. Most of the extragenic DNA sequences, 70 to 80%, are unique or exist as a small number of copies. The remainder 20 to 30% are moderately or highly repeated sequences that may be dispersed throughout the genome or lined up end to end as long tandem arrays. Dispersed repetitive sequences. Two types of dispersed repetitive sequences exist, known as short and long-term interspersed nuclear elements, abbreviated to SINEs and lines. SINEs. The best known example of SINEs is LU elements. These sequences are not identical, but they are similar enough to be classed as a family. They have an average length of 250 BP, and almost 1 million copies exist, which are very widely dispersed throughout the genome, occurring in most places, including the introns of some genes. The origin of LU elements is uncertain, but they are believed to have arisen as one or more processed pseudogenes. It is suggested that at one stage these sequences acquired transposable activity, which allowed them to replicate and move to different sites in the genome. Lines. They are similar to SINEs but have no longer sequences. A well-known example is the line L1 line, which is 6500 pp long and is present at 60,000 copies. Lines are a type of retro element. These are sequences that are capable of moving through the genome by a process called transposition. Transposition, which allows them to copy themselves by reverse transcription and to insert the copy into the genome at a distant site. Most L1 elements are truncated, but the full-length version has all the components needed for transposition, including a gene that could code for reverse transcriptase. Clustered repetitive sequences. The human genome contains extensive regions in which repetitive sequences are arranged end on end as long-term arrays. This is called satellite DNA and occurs in various forms, each with a different repeat unit, because they are separated from the bulk of cellular DNA by equilibrium density gradient centrifugation. However, not all the simple sequence DNAs separate from the bulk of cellular DNA during centrifugation. Similar sequence DNA, which consists largely of very short sequence repeated in long-term tandem arrays, is preferentially located in centromeres, telomeres, and specific locations within the arms of particular chromosomes where it may have a structural role. Three classes of satellite DNA have been defined based on the cluster and the length of the repeat unit. Satellite DNA that was the first type to be identified in the human genome and contains repeat units of up to 200 pp arranged in clusters between 100 and 5000 kbp. Mini satellite DNA occurs in small clusters up to 20 kbp in length and has repeat units up to 25 pp. The length of a particular simple sequence tandem array is quite variable among individuals in a C species, probably because of unequal crossing over during meiosis. Difference in the lengths of mini satellite DNA forms the basis for DNA fingerprinting. Microsatellite DNA. Microsatellite DNA occurs in short clusters, usually less than 150 pp, and has a repeat unit which is usually 4 pp or less. Microsatellite DNA is very common. One type, which contains dinucleotide CA as a repeat sequence, accounts for 0.5% of the entire genome. Mononucleotide repeats consisting of a single repeated base account for a further 0.3%. A variable number of tandem repeats. This will be explained in the next lecture. Take notes of these, and I'll be back with you in a short while from now. Just a general mention, the length of the microsatellite DNA sequences, such as CA repeats and mini satellite, varies from person to person.